Welcome to the video lesson on mitosis. In this video, we will discuss what mitosis is, the 5 stages of the cell cycle, what happens when the cell cycle goes wrong, how DNA appears in our cells at different times, and the 4 stages of mitosis. Let's go ahead and get started here by talking about the cell cycle. Your note-taking sheet has this pie chart, but you don't have the privilege of it being color-coded. So what I would encourage you to add the labels for each of the pieces of the pie chart here according to the color code on the right. This would be the G1. We'll talk more about that in a second. This is S, G2, M, and C. The G1 is called is the nickname for gap 1. This is where growth and normal cell processes occur, and most cells are found in this stage, gap 1 or G1. It is also important to note that this is where cell stalling occurs in this stage. So you should label this as well, as I've done so here in my diagram. In between G1 and S, this line represents the restriction point. And S is the synthesis stage. Here is where DNA doubles to provide each new cell a complete copy of itself. So synthesis means to build. So this is the synthesis, this is the building stage for DNA where DNA doubles. G2 is the gap 2 stage, and here we simply have continued growth and preparation for division. The M is the mitosis stage, and this is the division of the nucleus. The nucleus divides, the DNA is split exactly in half. And in the C stands for cytokinesis. And cytokinesis is the division of the cell itself. The cell cytoplasm divides in half, and we end up with what are called 2 new cells. These 2 new cells are called daughter cells. They are identical copies of what is called the parent cell. G1, S, and G2 are collectively called interphase. So these 3 stages together of the cell cycle are called interphase. M and C are the division phase of the cell, division stages of the cell. You will also hear this often called the mitotic stage, since that's when mitosis and cell division takes place. If a cell is stalled and begins to divide again, or if a cell begins to divide much faster than normal, cancer is the result. Some kinds of cancer begin when stalled cells re-enter the cell cycle, and the cell speeds up and becomes inaccurate, leading to masses of cells with the wrong chromosome number, and which are abnormal in other respects. There are 3 types of cell overgrowth. There are benign tumors, which do not invade the surrounding tissue, malignant tumors, which invade and destroy surrounding tissue, and metastatic tumors, which spread to other organs. Let's continue now by looking at DNA in the cell. DNA, of course, determines the physical makeup of an organism, and it exists in 2 forms depending on what stage of the cell cycle the cell is in. Chromatin is when the DNA is unwound. It appears tangled, and it appears this way during the interphase stages, again, which would be G1, S, and G2. The other appearance of our DNA is what are called chromosomes. Here, the DNA is tightly wound, thickened together, and appears this way during the cell division stages of M and C. Here we have a nice picture diagram of a chromosome, and a chromosome is made up of basically two parts. We have the chromatid, and this kind of looks like an X. So each half of the X would be a chromatid. So you have a chromatid on the right side and a chromatid on the left side. And then you have this protein center here, which is called a centromere. So the two key parts of a chromosome. Next, we're going to learn about what is called diploid number or diploid number, which is the number of chromosomes per cell. As humans, we have 46 chromosomes per cell, which is divided up into 23 pairs. You get 23 chromosomes from your mom, and you get 23 chromosomes from your dad, and they pair up to give you 46. Fruit flies, on the other hand, have just 8. Cats have 38. And if you think you're a big deal, dogs have 78 chromosomes. Pea plants have 114. And how about the maiden hair fern with a whopping 1,248 chromosomes? Clearly, chromosome numbers differ for different organisms. This is one reason why different species can't interbreed. The egg and sperm would have different number of chromosome numbers. Mitosis assures that each new cell has exactly the same genetic information. Next, let's take a look at the 4 stages, well, they're called phases of mitosis. We start off with prophase. In prophase, the nuclear membrane dissolves, the chromatin winds into chromosomes, the centrioles divide and move to opposite sides of the cell called poles and form what are called spindles. You will also hear these called microtubules. In our second phase, called metaphase, the spindle connects to centromeres at opposite ends, and the chromosomes are moved into the cellular equator, if you will. They line up in the middle. Students often remember metaphase because it starts with middle, M, and the chromosomes are lined up in the middle of the cell. Next phase of mitosis is called anaphase. Here the chromosomes split into chromatids, but recall that the two chromatids of a chromosome are identical, so no information is lost, and the chromatids move to opposite poles. Ana means back, or another way to think of this is away. A for ana, A for away. The chromatids are pulled away from the middle towards the end or edges, sides of the cell. Our fourth and final phase of mitosis is called telophase. You will also sometimes hear this called telophase. In telophase, the centrioles stop moving and become inactive. The spindles disappear, disintegrate. Two nuclear membranes reform, and the chromatids unwind back into chromatin. Telophase is followed by the cytokinesis stage, in which the cell continues and splits all the way in half into 2 new daughter cells. Next, let's take a look and have our 2 favorite amoeba sisters give us some wonderful animations on the process of mitosis.